The screen is visible, sir.
Yes,
Sir, visible.
visible, sir,
Audible.
Yes, visible.
audible as well as visible.
Hello. The screen is visible.
It is not visible now.
Okay, sir. Okay. इसको ऐसा इसको काट दे नहीं काटना नहीं सीधा पुट इसको now it's okay, sir.
Yes, sir, it's okay.
Okay, thank you, sir. As all of us are quite aware that the cell, which is a structural and functional basic unit of life, when it divides a large number of cells, numerous cells are formed. The cell which used to divide, that is parent cell or mother cell, and the cell which are being formed, that are daughter cells. When daughter cells are formed, this is cell division. Major characteristics of living organisms are shown by the cell growth and reproduction. Before starting the cell cycle, we must have these terms clarified to the students before the students, so that the students may get acquainted about somatic cells, germ cells, chromosome, chromatin, bivalent, tetravalent, haploid, diploid, cytokinesis, karyokinesis. It will be better if all of you will take part in each and every term, then it will be better. Somatic cells, that is growing cells. Germ cells, that is the reproductive cells. Chromosome, chromatin, these terms must be clarified before the students, before starting the topic in the classroom scenario. We must also clarify before the students that there are two types of organ cell, that is the prokaryotic as well as eukaryotic. Prokaryotic organisms will have single chromosome as well as they will have circular DNA known as plasmid. In eukaryotes, many chromosomes will be there. Prokaryotic chromosomes will be circular, whereas in eukaryotes, these chromosomes will be linear. in the prokaryotic organisms, chromosomes, they are only of DNA, but in eukaryotes, that is made up of chromatin material, nucleoprotein material, and coiled around histone protein. We can also find in prokaryotes the cytoplasm is there where we can find is DNA, but in eukaryotic cell we will observe in the nucleus, within the nucleus. Here the copies of chromosome, it divides immediately afterward, but in eukaryotes, copies, chromosomes, then the cell grows. Then it goes to mitotic cell division to organize the chromosome in two equal groups. DNA is located inside the nucleus of every cell in relaxed, uncondensed form, that is called chromatin. Chromatin is thread fiber, chromatin fiber is there, and DNA is the coiled form of chromatids that forms chromatids, and the loop of tightly coiled DNA, that is chromosome. Here we can find the chromosome structure, that it is the bivalent chromosome and this one is tetravalent chromosome. In the center there is the centromere, and both arms will have the DNA and chromatin fiber. When the both two chromatids unite together with the help of centromere, there is formation of chromosome, and the chromatids will be regarded as sister chromatids. One thing also should be clarified before the students, because when we are dealing the cell division, we will have to discuss homologous chromosome, sister chromatids, because without concept of sister chromatids, non-sister chromatids, they will not be going to understand about the crossing over. So we will have to give the idea. This one, here we can find two chromosomes are present here. Both are homologous chromosomes. Here homologous chromosomes are there, and the chromatids of each other, they are non-sister chromatids. Non-sister chromatids. We can find that the cell, when the cell used to divide, they used to form large number of cells, plenty of cells, and cell growth takes place. What happens when large number of cells are being formed? The differentiation leads to formation of tissue. Cells unite together to form tissue. Tissues unite together to form organ. Organs unite together to form system, and system unite together to form the whole body. The cell which divide undergo division, that is the mother cell or parent cell, and the form cells are daughter cells. Formation of new cells or the daughter cells from parent cell, that is called cell reproduction. And the cell division is a process by which parent cell, after replication of its component, is split to form daughter cell. And the cell cycle is the series of changes, series of events which occurs in a cell by which it duplicates its genome. Genome is duplicated. New genomes are formed. New copies are formed. And other contents are also formed by division into two daughter cells. The period required to complete one cycle is 24 hours in human beings or human cells, 90 minutes in yeast. This cell cycle term was given by Howard and Pele in 1953. Now, mitosis or mitotic cell division. Mitotic cell division we can find that was studied by Strasburger in 1870.
व्यवस्थित बोलते हैं लोगो डालने के लिए इसमें स्लाइड में ऊपर में लोगो होना चाहिए यहाँ पर सब स्लाइड में रख देना पड़ेगा ऊपर हाँ हर एक स्लाइड में डालना होगा
Strasburger in 18
बोला तो हम डाल लें करके हमरा कल देखेगा नहीं कुछ नहीं खाली प्रेजेंटेशन देखेगा और कुछ नहीं
Strasburger in 1870
सुजय का
and
का भी नेक्स्ट है सुजय का देखेगा हाँ
then it was studied by Bovary and Fleming in 1879. Mitosis is a type of cell division
लास्ट का
which occurs in somatic cells or vegetative cells where the cells are formed which are genetically similar to the parent cell. Means whatever the number of chromosome will be there in the
तो भरे हुए हैं
parent cell, that will completely in the daughter cell.
हाँ एकदम गोल
We can also find that as the number of chromosomes are same in the parent cell, hence it is also known as equational cell division. After the end of
देख रहा है हाँ हाँ ठीक
mitotic cell division, two daughter cells are formed. After the end of mitotic cell division, two daughter cells are formed and each daughter cell will have the same number of chromosome as the parent cell. Mitotic cell division has different phases, subphases, that is interphase, prophase, metaphase, anaphase and telophase. We can find that the interphase was previously regarded as the resting phase, but it's not like that. During this phase, we can find a lot of growth, development, metabolic activities are being governed. That's why it is called G1 phase. Duplication of chromosome DNA replicates, new copies of DNA are formed. That is called synthetic phase. Again, growth phase takes place. And then the prophase starts. Means before the start of this mitotic phase, prophase, this interphase has a lot of work to be done in the cell so that the cell components are being formed, the different types of substances are being formed, whatever substances which are required for the cell division that are being formed. After that, the prophase will start.
चालू हो गया ना
So that is the rapid growth where the metabolic activities
बुलाया जाता है
takes place, centriole replicates,
एक बार presentation
growth and development
आदमी इंजन
takes place, DNA synthesis occurs, and then growth phase again takes place. It means G1, that is the growth phase or the gap in some places, it is also called gap. Then synthetic phase, then again growth, and final preparation of the cell for the cell division. Then the prophase starts. As we have told that the interphase was previously known as the resting phase, but it's not like that. Cell grow in size, organelles are replicated, replication of DNA takes place, synthesis of protein occurs associated with DNA, synthesis of protein associated with mitosis, they are being formed. All these things happen within the interphase. Now, the mitotic cell division division of somatic cell in the eukaryotic organism forms two daughter cells having the same number of chromosomes as the parent cell, and that's why that is called equational cell division. Here the DNA replication takes place in the cell, and the cell undergo mitotic cell division, and two daughter cells are formed. Both are diploid. Both are having the same number of chromosomes as the parent cell. As we have told that the mitotic cell division has four phases: prophase, metaphase, anaphase, telophase. The students will learn that during the prophase, major events which used to take place in the plants and animal cells, that is going on, chromosome condenses, a spindle fiber forms. Chromosomes are captured by a spindle. Means these things are being formed, and there is the formation of. At the end of the prophase, we will find the cell membrane will be there, nuclear membrane will disintegrate, nucleolus will disintegrate, and mitotic fiber will start forming. But at the prophase ends, metaphase starts, the spindle fiber or mitotic fiber are clearly visible. We can also find that mitotic fiber or spindle fiber attaches the chromosomes, and the chromosomes are attached at equatorial plane. Means from both the poles, spindle fiber is observed and chromosomes are attached or arranged at the equatorial plane or at the center. This is the identifying feature of metaphase stage. If we will clarify before the students that this is the identifying feature, that presence of chromosome at the equatorial plane, students will learn that mitotic phase, metaphase will be understood or identified only with the help of the presence of chromosome at equatorial plane. After the end of this is the animal cell. Likewise, that we can also observe that in animal cell also there is presence of the chromosome at the animal, at the equatorial plate in the animal cell. Hello. Anyone wants to tell anything? Yes, Rama ma'am, mic open है. कुछ कहना चाहती हैं क्या?
Gajendra sir, should I say something?
हाँ कहिए सर.
At one point you were explaining about bivalent, monovalent. Sir, bivalents are tetrad.
आवाज सर आपकी कट रही है. आवाज क्लियर नहीं आ रही है. आवाज क्लियर नहीं आ रही है सर आपकी.
Am I audible, sir?
हाँ अब आ रही है.
सर मैं कह रहा था कि आप जिस समय बाइवेलेंट बता रहे थे, you were explaining bivalent means tetrad.
Bivalent means two. Bivalent means two. Bi means two. Tetra means four. So when both chromatids will join together with centromere, then it will be tetravalent. Hello, am I audible?
आप मेरी बात सुन लीजिए. मैंने what did I say? Bivalent means tetrad. Bivalent means tetrad. On either side of centromere, two chromatids are there. Presence of four chromatids. Two on either side of the centromere. Condition is bivalent, having four chromatids. But the term monovalent means dyad. One chromatid on either side of centromere. Is it clear, sir?
Yes, definitely bivalent.
Sir, there are two terms, monovalent and bivalent. monovalent, that is dyad, and bivalent means tetrad.
ठीक बोल रहे हैं
Yes, it's okay. आप कह रहे हैं अभी सही है, अभी सही है.
Okay, Gajendra sir, proceed.
ठीक है, okay. Now, after the end of the metaphase, students will learn about the how chromosomes are moving towards their own pole. Means movement of chromosome towards their respective pole will be going on. And when these things will be going on, the shape of chromosome will be V shape or J shape. And students must learn that yes, this is the identifying feature, this is the identifying character of anaphase stage. Spindle fiber attached to the kinetochores shortens two chromatids towards the pole. Free spindle fiber lengthen whose poles cell apart.
प्लस मांग कलर वाला कलर
Just after anaphase,
मैन पे यहाँ भेज चुका था सुबह
it comes telophase, where spindle fiber disintegrates
उसको बुलाया था उसके लिए
and nuclear envelope reorganized. Means whatever in the
ऑन करके हम कहते हैं
prophase stage, nuclear membrane was disintegrating. Now it will reappear. Reappearance will the things will be going on. Chromosomes revert to their extended state, and then cytokinesis occurs, enclosing each daughter nucleus in separate cell. Now, cytokinesis will be cyto means cell, kinesis means division. Division of cell. Cell will cut into two equal half. Plant cell undergo cytokinesis by formation of cell plate. It's very important thing to get clarified before the students that yes, you must know that how cytokinesis takes place in plant cell with the help of cell plate formation. In animal cell, it is with the help of cleavage or furrow. Means deepening in the both side, from the both side, deepening takes place. अंदर की ओर धंसेगा, and then it will cut off into two equal half. Then it will be cutting, and two equal halves will be forming into two new daughter cells. Now, the review. This is the image of mitotic cell division stages: prophase, metaphase, anaphase, telophase. So we will be providing this information, that interphase. Here just see prophase. In metaphase, at the centromere, at chromosomes are attached at the equatorial plane. This is the identifying feature of metaphase. V shape or J shape. This is identifying feature of anaphase, and cytokinesis is to form two daughters. This is about mitotic cell division. In animal cell, likewise that the same thing we will be dealing to the students, so that students must get acquainted about the plant cell as well as animal cell, because there is a great confusion among them that sir कैसे पहचानेंगे कि ये plant cell का cell division हो रहा है और animal cell का cell division हो रहा है. तो दोनों के सेल के स्ट्रक्चर में, दोनों के सेल के शेप को दिखा करके और हम लोग क्लियर कर सकते हैं. We can clear it. Now, the next one is meiotic cell division. Inside the body of the organisms, there is presence of two types of cell: somatic cell as well as reproductive cells. Reproductive cells are germ cells. They are the gametes or germinal epithelium or germ cells. In germ cells or reproductive cells, there is meiotic cell division. Meiosis is a type of cell division which occurs in the germ cell or reproductive cells. It's unique type of cell division. Here, the chromosome numbers are get reduced to half. whatever the parent cell has the chromosome number, that will reduce to half, and haploid formation will take place. Means we are diploid, germinal cells are diploid, but when germinal cell will undergo meiotic cell division, meiosis, they will form haploid cell, haploid gamete, and gamete will have single number of chromosome. So these things must be clarified before the students. Haploid, diploid, how chromosomes are, how organisms are haploid, which organisms are diploid, which parts are haploid, which parts are diploid. Now, this will help to get clarification before the students. We are having our parents, we match with our parents. So many characters in human beings as well as organisms match with the parents. Why this match? Why this thing happens? This is because of meiosis. This is because of crossing over. These concepts must be clarified before the students. Here, mother, father, 23 pairs of chromosome. Here also 23 pairs of chromosome. Here also. And when meiotic cell division takes place in both of them, what is happening? Gametes are formed having the 23 unpaired chromosomes, 23 unpaired chromosomes here also, and 23 here, from here, that is the male gamete, 23 from here, female gamete, X and X, or 23, both unite together to form the child or the baby, which is having 46 number of chromosome. In other words, haploid, haploid, diploid. We used to ask the students the question, अच्छा भाई बताओ, when the parent, we have our parents, what is the number of chromosome in our body? The students give to reply, yes sir, 46. What is the number of chromosome in the father? 46. Mother, 46. But when we ask 46, 92, how it came into 46 again? Most of the students do not give the correct answer. This will help to give the correct answer. This board will give the correct answer. Yes, 23 came from the mother after reduction division, that is the egg formation, and the 23 came from the father side, that is after the meiotic cell division, and the gamete formation, that is the sperm formation. So haploid, haploid, diploid, or 23 make the 46, and this is the child. Now, meiotic cell division is a longer cell division, which is divided into two phases, that is meiotic 1 and meiotic 2. Meiotic 1 will have prophase 1, metaphase 1, anaphase 1, telophase 1. Meiosis 2 will have also prophase 2, metaphase 2, anaphase 2, telophase 2. But the prophase 1 of meiosis first cycle is a longer cell division, and that is segregated or divided into 5 sub-stages, LZPD square. L stands for leptotene, Z for zygotene, P for packitine, D for diplotene, and D for diakinesis. We used to provide this thing LZPD square, और बच्चे को दिमाग में सेट करवा देते हैं. L से लेप्टोटीन, Z से जाइगोटीन, P से पैकिटीन, D से डिप्लोटीन, और D से diakinesis. Because it is not observed in my mitotic cell division. So prophase first having the leptotene stage, where we can find the chromatin begins to condense two sister chromatids so close, can't be distinguished. Here we can observe from the image, in the leptotene stage, the thread-like structures are being formed, homologous chromosomes are formed, but in the zygotene stage, both chromosomes come nearer, homologous chromosome come nearer, closer. That is, synapses used to take place. Closing nearer, that is synapses, zipper-like fashion. And we can also observe that just after zygote in a stage, crossing over takes place. In the crossing over, what is going to happen? Non-sister chromatids cross, overlap each other, and X-like band is formed. This X-like band is called chiasmata, and this feature is called crossing over. Here we can find this crossing over. Here we can understand this crossing over. Cursor is visible. Hello. Cursor is visible.
Yes, sir. It is visible.
Yes, sir. Yes.
Okay, sir. Just try to see. बच्चे को ही दिखाएंगे, बहुत अच्छी तरह से समझ जाएगा. Homologous chromosome. These are sister chromatids, but the chromatids of this one and this one, that is non-sister chromatids. Both non-sister chromatids overlap each other and cross-like band is formed. This cross-like band, that is called chiasmata formation, and this feature is called crossing over. And that we used to give the clarification before the students. that देखो यहाँ पे देखो, पैटर्नल क्रोमोजोम, मैटरनल क्रोमोजोम, बोथ क्रोमोजोम क्रॉस किया, और क्रॉस करने के बाद देखो एक थर्ड टाइप का क्रोमोजोम बना जिसमें पैटर्नल भी है और मैटरनल भी है. तो यहाँ पे देखो कि पैटर्नल क्रोमोजोम के साथ मैटरनल कितना है, कम है, 25%. तो पैटर्नल कैरेक्टर ज्यादा मिलेगा. If this chromosome used to move in the child, what is going to happen? Maternal chromosome will have more characteristics. So child will have more character. Child will have more traits matching with the mother. And if this chromosome will be moving, what will be happening? The parent's father character will be more and more in the offspring. So this image will be helpful before the students in the crossing over. This is chiasmata formation, and just after packaging, diplotin stage is observed. In the diplotin stage, we can find that the separation starts, means crossing over के बाद जो क्रोमोसोम क्रॉस ओवर किया अलग अलग होना शुरू हो जाएगा, diplotin, and dikinesis completely segregated. After the end of dikinesis, again the cell will undergo metaphase 1. In the metaphase stage, again the chromosomes will be arranged at the equatorial plate. These chromosomes will be different from the mitotic chromosome because in mitosis there was no crossing over, but in meiosis crossing over is clearly identified, and so that crossed over chromosome will be lying at the equatorial plate. But in the anaphase stage 1, we will again observe that the chromosomes will move towards their pole. Which type of chromosome? The chromosome which are crossed over. The chromosome showing the crossing over, that chromosome will move toward the pole. And the shape will be that again V shape or J shape. And after that, telophase stage, where two haploid cells are formed having X number of chromosome or N number of chromosome, which undergo meiotic 2. And meiosis 2 is just like mitosis, where from two daughter cells, 4 daughter cells are formed. 4 daughter cells are formed having the same number. What we have learned, what we have observed, that in meiosis 1, chromosome number get reduced to half. As the chromosome number get reduced to half, it is called reductional cell division. But what we used to observe in meiosis 2? Meiosis 2 we used to observe that the chromosome number remains same. So meiosis first is reductional cell division, meiosis 2 is equational cell division. That must be clarified before the students. Now, this was about the cell division, mitosis, meiosis, and now the regulation of cell cycle. As we are observing the cell division, one cell used to form another cell, how it is accelerated. We used to provide some chemicals, we used to provide some hormones, we used to provide some other substances to make the faster cell division, and that chemicals which when we spray, when we used to provide, the cell division increases, the rate of cell division increases. After the cell moves to the next stage of the cell cycle, the cyclin that were active in the previous stage are degraded. CDKs are the key regulatory enzymes involved in cell proliferation through regulating cell cycle, checkpoints and transcriptional events in response to extracellular and intracellular signals. Cyclin regulates the cell cycle only when they are tightly bound to the CDKs. To be fully active, the CDK cyclin complex must be phosphorylated at a specific location. In plants, cytokinin, a group of hormones that is called phytohormones. These are called plant growth regulator. Growth regulator because they regulate the growth, they regulate the cell division, and that's why they are called growth regulator. Phytohormones are derived from adenine, stimulate cell proliferation, that is cell enlargement used to take place, proliferation used to take place. Cytokinin signaling is initiated by membrane associated histidine kinase receptor and transduced through a phosphorylase system. Cytokinin signaling is initiated by membrane associated histidine kinase receptor and transduced through phosphorylase system. Cytokinin positively regulates cell division and also serve as a key role in establishing organization with suit stem cell center. We can find oxygen dependent and oxygen independent mechanism have been uncovered by cytokinin, which stimulates cytokinin, the endocycle in roots. The cyclin for the M phase CDK is only present in the M phase, mitotic cell division, interphase, mitotic, and again interphase. Means cyclin concentration is, here we can find cyclin concentration, how it goes up and CDK activity takes place. Means when it declines, when cyclin concentration goes up, when cyclin concentration is high, what is going to happen? CDK activity is observed. Means cyclin dependent kinase activity, cyclin dependent kinase activity is marked, is observed. That we will be also explaining before the students. Cytokinin promotes mitotic cell division and positively regulate a cell division. It also controls the cell cycle both at the G1 or the S and G2 or M transition, mitotic transition. Cytokinins are a group of naturally occurring plant growth hormones or phytohormones which are responsible for cytokinesis. Cyto means cell, kinin kinesis. It will be increasing the cell division. It will be increasing the rate of cell division. That is cytokinesis will enhance due to the hormonal concentration. Phytohormones, that is cytokinin. The hormones play a vital role in cytokinesis or cell division, the physical process of cell division. Here, the cyclin CDK complex are being formed. When the cyclin concentration increases, CDK are formed, and the dimer contains a regulatory subunit called cyclin, catalytic subunit called cyclin dependent kinase. This will catalyze the reaction. This will enhance the reaction known as cyclin dependent kinase. Now, formation of cyclin. CDKs control the cell cycle progression via phosphorylation of target gene such as tumor suppression, protein retinoblastoma, tumor suppressor, protein retinoblastoma. The activation of cyclin CDK is induced by mitogenic signals and inhibited by activation of cell cycle, the checkpoints in response to DNA damage. Cyclin CDK complexes, cyclin dependent kinases, concentration does not vary. No kinase activity unless they are associated with cyclin, means it is must. Cyclin must be attached to the kinase enzyme. Means activity will be unless associated with cyclin. Cyclin have no enzymatic activity, but when it binds to cyclin, binds dependent kinases to activate kinase. Cyclin dependent kinases to activate kinase. Their concentration in the cell vary in a cyclic fashion during cell cycle, means the concentration will enhance, concentration will decrease as per the cell. Here one image is there. Can you identify, sir, which type of image is here, which cell is here, which zone is here, which part of plant is here?
I think it's a root tip.
Apical meristem, sir.
Yes,
Apical
exactly. I just want to know from you there. Apical meristem, root apex is there, and this apical meristem will be showing here. Stem cell maintenance. Here also different types of zone is there. Different zones are here. Here cell elongation zone, cell elongation zone, cell differentiation zone, cell proliferation zone, quiescence zone. Which type of zone is here? Anyone can identify this one? Hello?
Meristematic zone, sir.
Yes, meristematic zone is here. Means cells are highly dividing. When we used to have the squashing of the onion root tip, we used to cut from this part and fix it so that maximum mitotic cell division may be observed, may be marked by even students also. So here you can find there are 3-4 activities: cell elongation, cell differentiation, cell proliferation, and quiescence stage. Here with the color it has been marked. Means in the cell elongation, there is the hormonal balance also. There is role of auxin, cytokinin, gibberellic acid, ABA, and it means joint. All these hormones work. Then the cell elongation used to take place. Cell differentiation and under endo-replication stage, we used to observe auxin, cytokinin, and gibberellic acid. These three used to work. And cell differentiation used to be marked. Cell differentiation used to be observed. Means here also there is hormonal role. Hormone play important role here also in the cell differentiation, cell regulation. Now, in the cell proliferation, there is also a role of auxin. GA, gibberellic acid, ABA, abscisic acid. ऑक्सीजन मार्केट में किस केमिकल के फॉर्म में मिलता है या किस केमिकल के नाम से मिलता है सर? हेलो, ऑक्सीजन का केमिकल फॉर्म क्या हुआ? सिंथेटिक ऑक्सीजन जितने भी है, सिंथेटिक ऑक्सीजन,
Indole acetic acid. That is natural oxygen.
Yes, right. Foundation is right. Indole 3 acetic acid, IBA, indole 3 butyric acid. These are the synthetic oxygens. Then we can find that after cell proliferation, where the gibberellic acid, oxygens, and ABA plays important role, then we can find that when the asymmetrical cell division of stem cells used to take place, there we can observe the auxin also play role. Auxin also play pivotal role in the cell differentiation, cell elongation, and that's why it used to also show the apical dominance. We must clarify before the students that why the cell division is marked here due to concentration of auxin, and this concentration of auxin will proliferate more and more, and they will form large amount of cells, and that's why cell elongation used to take place. The height of the plant used to take place due to this hormonal activity, hormonal concept that we will also provide before the students, that yes, you can understand, you can find that these hormones can play a very important role in the cell division. If the plant's growth are not being marked in your garden, please try to use these hormones, and when you will be using, the plant's growth will be observed. Sometimes we used to find in our garden also, flowering is not being observed, stem height is stunted. Then we used to just provide the hormone, spray the hormone, and the cell activity is marked. Then proliferation is marked, then new branches comes out. And what are these? These are the hormonal regulation, hormonal conceptual understanding among the plants, within the plants. What is the role of hormone in cell division? What is the role of hormone in cell cycle? Now, revision and home assignment. Explain mitotic cell division with the help of diagrammatic representation. Explain why mitosis is regarded as equational cell division. Explain different stages of prophase first of meiosis with the help of suitable diagram. Explain why meiosis first is reductional cell division. Explain why meiosis second is called equational cell division. Then explain what is crossing over. Enumerate events of crossing over with the help of suitable diagrams. And finally, after the end of the complete period or the chapter students will be able, all of you know, but students will be able to give answer about the cell cycle. Now they will have no problem to give answer about the cell cycle. What is cell cycle? They will frequently give the answer. They can give you what is growth, what is equational cell division, what is reductional cell division, what is crossing over, why do we have some traits matching with our parents, role of hormones in cell division. Means all these things students will be get acquainted after the end of the chapter. This is all about my presentation. Now, as yesterday sir was telling that it is too big topic, how it will be done. So it has been done. Now it's your time. Ma'am, as I was being given 1 hour time to just finish so that the proper discussion should be there. So now it's your time.
Thank you, Gajendra sir. It was a nice presentation, beautifully presented. I have touched almost all the topics, sir. But the only thing is how the students should be able to learn in an effective way. किस तरह से उनको रोल प्ले या किस तरह से उनको कराएं, कोई एक्टिविटी कराएं जिससे ये सेल डिवीजन के प्रोसेस को अच्छी तरह समझ जाए. Most problem comes from प्रोफेज 1 स्टेज. प्रोफेज 1 में जो 5 सब स्टेजेस हैं, दे फाइंड वेरी डिफिकल्ट इन लर्निंग. निमोनिक्स तो ठीक है, निमोनिक्स से याद आ जाएगा, बट द प्रोसेस, द कॉम्प्लिकेशंस देयर, द स्टूडेंट फाइंड डिफिकल्टी देयर ओनली. एंड दिस कैन बी डन.
वीडियो दिखा सकते हैं, वीडियो देख सकते हैं.
ये सब बच्चों के द्वारा कुछ जैसे वुलन प्रोडक्ट्स हैं, ऊन है, उससे वो कर सकते हैं बाय पेंटिंग ऑल दो.
लर्नर के लिए वी आर यूजिंग वीडियो. लर्नर के लिए वी आर यूजिंग दिस प्रेजेंटेशन इमेज फॉर्म. लर्नर के लिए स्पेसिंग करके दिखाएंगे. यूज्ड टू शो इन द क्लासरूम एंड मॉडल्स आर देयर. उसको दिखा करके स्लो लर्नर के लिए समझाना वही एकमात्र रास्ता है.
यस सर. और लोग कुछ कमेंट करना चाहेंगे? एनीबॉडी?
Am I audible, sir?
Yes, sir.
Yes, you are audible.
Sir, when we start explaining crossing over,
इसको हो रहा है सर. 2 डिवाइस एक जगह है, बंद कीजिए इसको.
When we start explaining crossing over, we should highlight 3 points at least: recombination nodule, recombinase enzyme, role of recombinase enzyme, chiasma, etc. We must focus on these terms.
Yes, only then they will understand. Yes.
Sir. Recombination nodule, role of recombinase enzyme, at which stage? At late pecadin. As we know, crossing over starts at late pecadin and is completed at diplotin. We must take to our students. Alok sir.
Yes sir, yes sir. I'm excuse me sir, for 2-3 minutes I am busy with some important official work, sir. Please
Okay.
continue.
Gajendra sir,
Yes, sir.
am I right?
Yes, 100% right. Without this word, how students will come to know that why paternal characters used to come in the offspring. It's the only one.
Total सुधीर सर की है.
Nice.
ये है मैडम का.
जब हम स्टूडेंट को एक्सप्लेन कर रहे हो मियोसिस स्पेशली रिडक्शनल डिवीजन, तो उन्हें ये भी क्लियर करना होगा कि हाउ क्रोमोसोम नंबर इज रिड्यूस्ड टू हाफ, एट विच स्टेज this one is responsible, how it gets reduced, how chromosome number reduced to half, which stage is responsible, how is mitotic prophase different from meiotic prophase, how is mitotic anaphase different from meiotic anaphase. If we are in a position to differentiate mitotic prophase, meiotic anaphase, meiotic anaphase, then I think we will be in a position to explain the main point.
Yes, sir. Exactly true. 100%.
साइंस में लिखा हुआ है।
हाँ that's science है।
हाँ तो वो साइंस का हो गया तो कॉमन कर दीजिए।
Kavita ma'am, are you agree?
Yes, sir, definitely. You are right. These fundamental things need to be clarified also. And then we can proceed further. That is true.
अगर ये ठीक नहीं लगता है ना
That was in the recapitulation part. In the recapitulation part that I have given the first slide, that the students must get acquainted with all these terms. If they will not be understanding these terms, the cell cycle should not be started. करना ही नहीं चाहिए, करना ही नहीं चाहिए.
कोई था ही नहीं ऐसा नहीं
Sir, in cell cycle, especially when we talk about synthetic phase, we must say about splitting of centriole. Division of centriole occurs especially at synthetic phase.
Yes.
This point must be clear before the students. Apart from DNA replication, division of centriole is very, very important.
Yes, yes.
अच्छा हुआ ना अपने से आ गया ना
Very true, sir.
ये हम अपने से डालें 11th का जो दूसरा को बचा है ना ये हम खुद से डालें ना। हाँ
सब करेंगे
हाँ हम देख रहे हैं
Any query from my learned colleagues?
क्योंकि 11th का पार्टिसिपेशन बहुत जरूरी था सर।
Ravi sir,
क्योंकि भविष्य
Yes, it is joint venture. Please share your views.
Ravi sir, please share your views. Ravi Kumar sir.
उसमें भी 11th का पार्टिसिपेशन बढ़ाना हाउस कैप्टन वाला हाउस वाला कुछ हो जाता है।
हाउस वन ना छोड़ दीजिए उसको तो
Sir, the session was excellent, and I've got many things to know about the cell division, and that's all I want to say.
Devrat sir, please comment.
The way Gajendra sir explained, that is a nice one. Actually, when we are in the classroom situation with chalk and board, we use different colored chalk and try to show the process of crossing over. And with the help of that, as Gajendra sir suggested and shown, we also explained the same. But so far synaptonymal complex formation is concerned, as you are rightly said, sir, that during crossing over, before that, we must explain these processes, that recombination nodule, how it forms, the role of recombinase enzyme, as well as chiasma formation.
Actually,
But
sir,
whatever Gajendra sir explained, that is okay, and very nice presentation, I should say.
Actually, sir, pairing of homologous chromosome is known as synapsis, and this is caused by a special complex protein, that is synaptonymal complex, which gets dissolved after diploidin stage. It persists up to diploidin.
Actually, we must say all this to the student
Okay,
so that they will understand properly.
right. Vivekanand sir, Vivekanand, principal sir, would
Yes.
you like
Yes,
to
sir.
highlight?
Okay. lesson is almost up to the mark.
Actually, he tried his level best.
Yes, yes.
Aparna ma'am,
Yes, sir.
please come in.
Yes, sir, sure. As usual, sir has presented very nicely and effectively. I just want to ask one term, sir. Can he explain it again in a much easier way so that as we will explain to the students in that way? The term monovalent and bivalent, sir.
Yes, sir. Yes, ma'am. Actually, crossing over occurs at bivalent state. It occurs at tetrad. Bivalent means two pairs of chromatids. One pair of chromatid on either side of the centromere. If chromosome had four chromatids, two on either side of the centromere, the chromosome is said to be bivalent chromosome. And this is called tetrad stage. As we know, crossing over takes place between non-sister chromatids of the homologous chromosome. We know that it occurs between the non-sister chromatids of the homologous chromosome. So, the student must know about monovalent and bivalent.
Yes, sir. Thank you.
Gyan sir,
Yes, sir.
would you like to say something?
Yes, sir. Presentation was very good. I think we should must share with our student regarding the mitotic poison. That is colchicine, I
What is
think.
about colchicine? Colchicine.
Yes, sir.
It's an alkaloid. Colchi or colchicine is an alkaloid which helps to break the spindle fibers. If we treat mitotic stage with this chemical, there will be doubling of the chromosome. DNA number gets double. So actually, this is used especially in polyploidy. This is for competitive exams, sir. I think this is for competitive exams. Colchicine is an alkaloid which is used to break the spindle fibers. यदि स्पिंडल फाइबर ही ब्रेक हो जाएगा तो आप जान रहे हैं क्रोमोसोम के कैनेटोकोर से अटैचमेंट नहीं होगा. वो अटैचमेंट नहीं होगा तो आप जान रहे हैं वो अपोजिट पोल्स में जाएगा नहीं. तो नंबर जो है वो डबल होते जाएगा. Mandal sir, DK Mandal sir.
Yes, sir. Gajendra sir has explained very nicely, and it is very fruitful. In our in the class 11 syllabus, there is no regulation of cell cycle. Although we have learned a lot about it, and I want one thing. That I want to say something about the role of centrosome should be explained properly among the students, because in animal cells centrosome is there, but in plant cell there is no centrosome. So how cell division takes place in plant cells? It should be explained properly, I think.
Cytoplasmic proteins are there.
Yes.
Cytoplasmic proteins are there.
I think it should be properly explained among the students, otherwise it will be confused.
Kavita ma'am, please come in.
Thank you, sir.
हाँ सर.
कुछ बोले मैम.
हाँ जरूर सर, सही बात है. इसका बहुत सारा एस्पेक्ट्स है एक्चुअली, जो कि आई थिंक इन 1 hour it is difficult to take all the aspects and all the factors properly. So if certain topics are picked up, I think it will be better. ये थोड़ा vast हो जा रहा है.
Entire chapter cannot be explained in 1 hour. Neha Mitra ma'am, please come in. Neha ma'am is present or absent? Neha ma'am from Hazaribagh. Ma'am is present or absent?
Hello. Can you hear me, sir?
Neha ma'am, please comment on the topic delivered by Gajendra sir.
Yes, sir. Am I visible, sir?
Yes, now you are audible.
Yeah. Actually, I was having a bit network issues. That's why I was not able to switch on my video continuously. Yeah. Actually, the problem that I face when I teach in the class is generally, in order to make them understand many phases, first of all, I'll talk about that presentation by Gajendra sir. Obviously, it's very good, very nice. But from my point of view, like what problem I face in the class is that when I'm explaining things about like what is interface and all that stages and all that, they face a lot of problems, specifically when I'm discussing about the bivalent tetrad formation and all that, and the important stages like this leptotene, zygotene, liptotene, and all that. So though after 2-3 classes they are able to understand, but is there any, since all the teachers are present over here, is there any specific and easy way to make them understand this just at once?
And in classroom, by making proper diagram, the student will be in a position to explain the thing only if they are in habit of making proper diagram. If they understand the diagram, they will explain it.
Yes, sir. I do that. So this is what I faced once or twice. I faced this problem, but yes, eventually they understand. And after that, whenever we are using these smart board or other than videos have been shown, then it's clarified. Then up to some extent.
There are some videos also that
Yes,
will
sir,
help.
yes, sir.
That will help.
Animation, sir. Animation. Best way to teach
Animations mostly,
animation.
Yeah.
Yes.
Kavita ma'am, Pragya ma'am is there. Coordinator ma'am.
Pragya ma'am. So how was it? Ma'am is there. No one followed. I went at the almost at the end.
Provide us Google form. Okay. Thank you, ma'am.
When I was going on the way, I shared the link if I get late because I was on round to all the other subjects. Okay. The link has been shared, and you can give your reviews and fill the assessments. How was the assessment for the last one? I was getting very scared that I'll get wrong because it was a little bit confusing. Some of the questions about the opera. 2 questions I found difficult. I don't know if I did it correct or not. Discussions are about?
क्या पता। हाँ 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 है